And let's take a look at scientific discovery. So we got 4 questions here for the beginning of the video. These same questions will show up at the end of the video, and then you kind of can test yourself out, make sure you know what's necessary for this little section. What did Chargaff's analysis of the relative base composition of DNA prove? What did Griffith find in his work with pneumonia-causing bacteria and mice? Southern blotting, what is that? And microscopy techniques in the late 1800s, what did that enable? So those are good questions for this section. So we'll go ahead and try to cover that stuff. Okay, so in the correct chronological order, starting with the first scientist to make a contribution to our understanding of the structure and function of DNA, would go in this order that we have here. So it would go in the order of Griffith, Avery, McCarty, and MacLeod, followed by Hershey and Chase, then Watson and Crick, and finally Meselson and Stahl. So we'd like to know that order in terms of the discoveries made around the structure and function of DNA. So we'll jump into each of these guys a little bit. So we'll start off with Chargaff and his first and second parity rules. Through careful experimentation, Chargaff discovered 2 rules. Those are called Chargaff's rules that helped lead to the discovery of the double helix structure of DNA. The first parity rule was that in DNA, the number of guanine units is equal to the number of cytosine units, and the number of adenine units is equal to the number of thymine units. This hinted at the base pair makeup of DNA. The second parity rule was discovered in 1968. It states that in single-stranded DNA, the number of adenine units is approximately equal to that of thymine, and the number of cytosine units is approximately equal to that of guanine. He also observed that the relative amounts of guanine, cytosine, adenine, and thymine bases vary from one species to another. This hinted that DNA rather than protein could be the genetic material. In his work with pneumonia-causing bacteria in mice, Griffith found that some substance from pathogenic cells was transferred to non-pathogenic cells, making them pathogenic. In his transformation experiments, Griffith observed that mixing a heat-killed pathogenic strain of bacteria with a living non-pathogenic strain can convert some of the living cells into the pathogenic form. And real briefly on Edwin Southern, he was a British biologist and he developed what is known as southern blotting, which causes DNA sequences to be transferred to a membrane and identified with a probe. That's really all we need to know about Edwin Southern. And then really briefly on mitosis and meiosis as far as actual scientific discovery is concerned. The improvement of microscopy techniques in the late 1800s allowed the study of meiosis and mitosis, revealing parallels between behaviors of genes and chromosomes. This process of meiosis and mitosis are crucial in our understanding of genetics. And then we have 2 more here. So we got Thomas Morgan and then Sturtevant. When Thomas Morgan crossed his red-eyed F1 generation flies with each other, the F2 generation included both red and white-eyed flies. All the white-eyed flies were male. The explanation for this result was the gene involved is on the X chromosome. Females have 2 X chromosomes, of course, while males have 1 X and 1 Y chromosome. Morgan and his colleagues worked out a set of symbols to represent fly genotypes. The genotype of a vestigial fly must be VG/VG. This is the only option that contains the correct genotype for a fly. And then finally, Sturtevant provided genetic evidence for the existence of 4 pairs of chromosomes in Drosophila by showing that their genes cluster into 4 distinct groups of linked genes. Okay, and then we get back to our questions. So that's kind of a short little video on some of the stuff that we need to know about scientific discovery. And one of the best things you can do now is hit that pause button and make sure you can answer these questions.